Thank you very much for the invitation. It's a real pleasure to be here. Thank you for the invitation, and thank Angela for connecting with me a couple of years ago and connecting me with Patty. And I spent a nice day yesterday in Boston talking about gene therapy approaches and how we might use that. I sent it on to you, yeah. So, can I move this on, or you?
You should be able to. Oh, there's a remote, right?
Oh, yeah. I come back on you. We go. Okay, so a little bit about me, because I'm a slight outsider. I've worked for 25 years now on vascular disease, and twofold. One is to understand smooth muscle cell function, and that's been in relation to vascular injury, usually quite commonly in acute vascular injury in the context of vascular grafts, etc. Spent a long time working in those settings, but also very interested in gene therapeutics broadly. I sat on a UK Gene Therapy Advisory Committee for a decade, and that was involved in making ethical and scientific decisions on all gene therapy that happened in the UK. I brought those together, and we've tried to develop gene therapies to target the vasculature modifying smooth muscle cells in the context of these common conditions. So this is not, of course, an MSMDS, but I think there are parallels, and I think it's useful to understand in a broader context. So I've worked in the UK a lot. Throughout that, I'm head of the cardiovascular centre in Edinburgh. And throughout that experience, I've worked in different places where we can sort of build on different aspects of smooth muscle cell biology, as well as bringing some of the technology they require together to form gene therapies. I've also spent a lot of time working on viruses that we can use as therapeutics for genes. So it's really essential to understand how you can deliver genes effectively. I think if you can get to a smooth muscle cell, you can quite easily modify it. It's relatively straightforward, but getting something to that cell is quite difficult to achieve. So I've worked with companies such as Janssen in their vaccine program, where they can develop vaccines based on viruses, and we use them for gene therapy, and that's been something we've been doing for many years. Next slide.
You should
show me how to change out.
Yeah, the remote in there.
Okay, so if we start, let's just go back to exactly the first study that showed vascular gene transfer. So that was over 30 years ago, from Betsy Nabel in 1990. And this is where taking a section of blood vessel in a pig, and this is not about ability to inject genes or viruses that can then home into the vasculature. This is using a catheter with a double balloon that you can inflate, and then you can infuse something into the middle. That allows a bit of exposure time for the blood vessel with the gene delivery system, because you need that contact to allow uptake of that gene delivery system into the vascular cells. It doesn't really happen until recently, and that you can inject systemically and you'll get the vascular to take up the tissue. Okay, so originally all of the applications were about either direct injection into muscle or using localized injection when you can have some dwell time that allowed that delivery to take place. And then there were a whole slew of sort of first in human gene therapy studies injecting into the muscle, and most of these were in trying to generate new blood vessels. And using vascular endothelial growth factor, angiogenic agents, so agents that promoted blood vessel growth for patients who had either myocardial or peripheral ischemia. And that concept is quite good. You can deliver directly into the tissue. In animal models, you can grow blood vessels, but this never really worked in patients. It was always too difficult. And actually, when you look back retrospectively, I think that was quite negative for the field, because this is not proven to be efficacious, and there are no medicines based on this that have come forward, despite probably 25 years of research. So not everything works. Of course, the big game changer for the whole field was this, which was for an ultra-rare condition, lipoprotein lipase deficiency using an AAV. This was called Glybera, which is direct injection into the muscle using an adeno-associated virus, which is a virus that's being used a lot for gene therapy these days. Now, the data to support this wasn't particularly good, and it's not, the license hasn't been maintained, but this was the first licensed approved medicine in the Western society. And it really broke the back of the regulatory system to say we can approve gene therapies. And despite the lack of data in this and the lack of the license being continued, there's been a lot of developments over the last years of gene therapies that are really made it to FDA and EMA approval. They're mainly in rare and ultra-rare genetic diseases that are non-cardiovascular. So when you look back at the field, there's been great success where you can deliver genes effectively into the organ or organ systems that you need to correct the deficiency. So a lot of this has been in stem cell gene therapy, where you have access to the stem cells ex vivo. You can take them out, you can transduce them with a viral vector, and then you can put them back into the patient. Or in hemophilia, for example, where you can inject a virus that naturally homes into the liver. It's very, very effective. And then you can produce factor 9, factor 10, factor 8, whatever vector. And they've been very successful gene therapies. So the context is that if you can deliver to the tissue, then you have a chance to have an efficacious therapy that then makes it through to licensing. But none of them are in cardiovascular. And as I say, delivery is really the sort of key for gene therapy. So, of course, for MSMDS, we're talking about systemic gene delivery. That's very difficult. It requires a lot of precision. It requires a lot of advancement of the technology to make sure that can happen. In the heart, again, very, very difficult to deliver genes into the heart. And a lot of current gene therapy clinical trials are using intravenous injection, where actually the virus doesn't really home into the heart very effectively. So for me, that's pushing the system a little bit too far and it's very difficult. So there are techniques where you can deliver things locally using catheters, etc., where you can advance directly into the heart and put a needle into the left ventricle, or use perfusion techniques that sort of drip the virus from a catheter that's in the coronary arteries. That's more effective about localizing the gene therapy in the right place. What we've done is just try and hedge our bets a little bit, and at the bottom there is actually do an ex vivo gene transfer for patients who are having bypass grafts. And we think that's been most effective because we have access to the tissue, and that's always been very important for us. So I'll just tell you a little bit about the how that's developed. So patients who are undergoing bypass grafts, of course, generally elderly, multivessel disease in the heart, and of course trying to bypass an occlusion in the coronary vessels. And without that, of course, patients have angina, myocardial ischemia, and may infarct and have the consequences of that. A very common procedure, of course, and there are 1 million cardiac and 1 million peripheral graft operations every year, is to take saphenous vein from the patient's leg, which is a nice bit of material, and use that to bypass, usually coming from the aortic arch and then distal to the occlusion in the coronary arteries. So that is a very effective procedure. 100% of patients will have an immediate response in terms of their blood flow into the heart. So the myocardial reperfusion is really effective. The problem with this is that 50% of the vessels end up looking like this. So on the left-hand side, you see sort of a gross picture of a bypass graft that's failed. Okay, so what you'll find here is essentially you have what's called neointimal hyperplasia. So this is where the blood vessel wall has been damaged. You put a vein into an artery, it has to arterialize itself, so it expands, and that's really important, but it also has damage on the luminal side. That activates all of those smooth muscle cells. Okay, so it creates damage to them. And in a normal blood vessel, of course, the smooth muscle cells are generally quite quiescent. They don't proliferate much. They don't move apart from to organize vascular tone. The problem in this setting is the higher blood pressure leads to that vascular damage. And that leads to, in 50% of cases, which is a major clinical problem, leads to late vein graft failure. And on the right-hand side, you can see a histological image of what that looks like. It's occlusion. So actually what happens is the smooth muscle cells become damaged. They migrate into the lumen of the vessel, and that leads to this sort of pro-atherogenic environment. And you get a very, very rapid atherosclerosis. And that leads to complete occlusion. The graft fails, the patient's back in or has a myocardial infarction. And going back into these patients is very difficult. So we've always thought about, well, if you can do something at the time of surgery, would this be a useful thing? Because at the time of surgery, you have access to the tissue. Can we do something to prevent that happening? That's always been our hypothesis. So we can do this quite easily. This is not going to be a systemic therapy because we want to target things that have to have a local activity, because I think there'll be a lot of side effects. And it has to be delivered via gene therapy. This is not something that you can do with a small molecule. So what we did was try to understand, spend a lot of time on the basic science and trying to understand how this response to injury could be modeled and can we understand it. So very simply, in a healthy vein, which is what you start with in a bypass graft, you have the smooth muscle cells that are really maintained their Factor 2 positive. They have high levels of the microRNAs that Mark talked about earlier on. They're really healthy. They function well. And they're surrounded by a matrix. And this sort of matrix is a cage around these cells that holds them in that formulation. When you put them into the arterial circulation in a bypass graft, all of that goes. Okay, so you have that sort of physical damage by the elevated blood pressure. You have damage to the vascular wall. And this matrix is digested by a family of enzymes called metalloproteinases. So they become expressed in those cells, they're secreted, they chop away at the matrix, and those cells start to move, they start to proliferate. So they move and form this luminal damage that you see. So we thought a very easy way of doing that, to prevent that, would be to create a gene therapy that would block that enzyme activity. Unfortunately for us, these enzymes have genes that are natural inhibitors. They're called tissue inhibitor metalloproteinases. And even better, there are only a couple of them, and they inhibit lots of MMPs, each of them. So each of them has an effect across a broad range of these enzymes. So we thought, why don't we just shift the balance? Why don't we make some viruses, overexpress the inhibitors, and see whether we could do something useful? So how were we going to deliver that? Well, we had, at the time when I started this, which was a long time ago, we only had the choice of one virus, really. And this was pre-pandemic. This is 20 years before the pandemic. So we used adenoviruses, not adeno-associated viruses. And Matthew will talk about those separately, but these are adenoviruses. They're quite big. They're very common. They're only associated with very mild disease, so respiratory disease, ocular disease. They're self-limiting to a patient who's immune competent. In immune compromised situations, and particularly on kids undergoing bone marrow transplantation, they can be a bit more pathogenic, but largely self-limiting. And it doesn't matter about this below, but it has a very complicated genome, but we know lots about it. And it's easy to make a recombinant adenovirus. So that's what we did. So you can just change the genome a little bit. You can take out the parts that make it replicate, and you can put in your transgene, so our TIMP gene into that. And the advantage now is, of course, everybody understands a little bit about adenoviruses, because two of the vaccines in the pandemic were adenoviruses. Tens of millions of patients have been injected with these. We use an adenovirus called 5. The AstraZeneca virus that was used in the pandemic was a chimpanzee version. essentially exactly the same. So they're well known and understood and not really associated then, of course, with complications when they're given to the human population. So how do we do this delivery? So what we said we worked with the surgeons a lot and said, well, okay, we do this ex vivo. You take the vessel from the leg, and we have it. And what happens generally in bypass surgery is it's harvested first, of course, and then it's left in heparinized blood or saline for a period of up to 30 minutes. And we said, okay, can we use that time to do something with it where we can take our virus and administer it to the graft? So we do this, we can cannulate the blood vessel. It takes, I don't know, about a minute to cannulate it. And then we can just infuse with a syringe, super simple, into the lumen of the vessel. We let it sit there for 30 minutes. And then the virus interacts with the blood vessel wall and gets into those cells. And it's a very efficient procedure. Okay, so on the right-hand side at the top, so when we've taken, this is human tissue, the human saphenous vein, we can, there's excess to surgery. We can take that back to the lab. We can do this procedure, and then we can keep that alive for a couple of weeks. So on the right-hand side is 7 days later. You're looking at the lumen of the vessel, okay, looking down on it because we've opened the vessel up. And you can see any cell that's blue has taken up the virus and is expressing it. So this is a super efficient procedure. So maybe 50% of the cells at that surface are infected by the virus. So this was a very simple way of not changing clinical practice and getting a very efficient gene therapy into the vasculature. And this works rather well. We've done this in the human model, which you can keep alive for 2 weeks. That's great because it's the actual tissue we would use clinically. We've done it in a pig model where we can take an autologous vein from the pig's leg, we'll put the graft in, do the gene therapy. You'll see here again, this blue coloration, this is the artery. There's no blue coloration here because this is a very localized gene therapy. And you'll see on the other side here that on the far right, there's a control vessel that hasn't had any virus or has had a control virus that doesn't express the gene. And you see this pathogenic remodeling. The lumen's quite small, the neointima on this inner surface, which is full of smooth muscle cells that are proliferating, migrating. That's the pathological remodeling that you see in vascular grafts. And you can see when we overexpress our inhibitor, there's a clear difference between this. So this works really well. We're targeting smooth muscle cells that become pathogenic by preventing that process. And that prevents the whole pathological remodeling of the vessel. So to cut a long story short, over a period of a long time, we've developed this concept. We've done pig studies that take this out for a month. We've done pig studies that take this out to 3 months to show that actually when you just do this 30-minute stuff in cardiac theatre, that you get a long-term benefit. We wrote grants to the UK government and the British Heart Foundation. They gave us a lot of money to do a clinical study. We spent quite a long time manufacturing a clinical grade virus. It's very difficult, manufactures whole science in itself. We did another pig study to prove that the clinical batch was efficacious. We've done the health economics, which is important for if we're going to do this as a more broadly, if it's effective, to say, could, you know, if this was a business, could you build on that and sell this to the healthcare systems? We've done maybe 100 documents for the clinical application in the UK to make sure we have regulatory approval. And we've trained the hospital system to take the virus in, keep it, store it, deliver it to cardiac theatre, and everyone trained in how to do it. And that's been a long process, but an effective one. But you can understand maybe how long that takes. And doing it academically rather than through a company is actually quite challenging. The only way to do that really effectively is to build ecosystems where you have everything together. So there's a really terrific here that the sort of patient alliance is at the center of that. It's super important. My experience on the Gene Therapy Advisory Committee, which I'm going back 20 years, the UK was not very clear about whether it was going to do gene therapy for haemophilia, because it wasn't sure there was a real clinical need. So the patient voice, the patient advocacy agencies were really pivotal in making that decision towards advancing first-in-human gene therapies for haemophilia, and that's been super successful. They're all licensed. So bringing all this together in a organized way, in this ecosystem where you have that, you have the scientists, you have the manufacture, you have the animal modeling, you have health economics, etc. all together was really super important. So we've built this. This trial is going to be open in a couple of weeks. This says mid-April, but it's been delayed, so it's going to be mid-June. But we've developed it. We'll see how it works. Safety study will take a year. Then a phase 2 study would be in 120 patients. A phase 3 study would be in about 500 patients. But we're not funded to do all of that. We'll be funded to do some of it. So we'll see whether that works and is effective. But there's quite a lot of messages about how effective vascular gene transfer can be if you stack things in your favor in terms of the delivery systems and the mechanisms that you use to modify the smooth muscle cells. This is the projectile. It's a vascular gene therapy. It's brought together a lot, the government, the healthcare system, the funders, the charities, I say patients, and the whole healthcare system under one umbrella. And that's starting now. I think just to finish, a couple of things to note. I think what I'm using now is a virus that I made maybe 20 years ago, because it's taken so long to do that. And there are so many improvements in viral vectors and non-viral vectors that can now, as Patty has developed an AAV vector that will deliver to the vasculature when you deliver it systemically. But we've taken something that developed a long time ago. At some point, you need to lock it in and develop the clinical environment around it to allow that evidence base and the regulatory and the manufacture all to deliver that virus. We have continual improvements, but they take a long time to get back into the clinical pipeline. There are always companies, academics interested in developing viruses that target different systems, targeting the heart, targeting the cardiovascular system. So I think this is a really important academic area where we need to make sure that governments and companies fund this, and we're working actively on that, as well as how you improve expression and make it very selective for the cell type that you're interested in, despite the fact that some of the viruses will go to lots of different tissues when you inject. What I can also do a little bit, because I'm slightly arm's length from what you're doing here, but I'm very happy to help from a UK side. Just to say that I think in the UK there could be a lot of connectivity. The UK has invested very heavily in advanced therapies. A couple of examples for you. So the British Heart Foundation invested 30 million in trying to find advanced therapies for cardiomyopathies, so inherited cardiomyopathies. That's going extremely well, and that builds those ecosystems across the world. These are funded in the UK, but globally, with all the leaders in the world, with industries to bring it together to develop cardiac gene delivery. And the Medical Research Council, which is the UK funder, has just announced in the last 6 months 2 centres in advanced therapies for 100 million. That's massive funding from the government. One is in cardiac therapies, which I'm co-directing with a couple of colleagues of mine. And the other one, which is quite interesting, is in an MRC, it's a Centre of Research Excellence, 50 million, run from Oxford, which is in rare genetic diseases. So this is therapeutic genomics, and it's particularly focusing on areas of rare diseases, rare disorders that cause severe seizures in infants and neurodevelopmental delays. And it's about bringing together communities that are working in very similar but different rare genetic diseases and developing therapies on scale. So bringing the cost of those things down and making them effective with bringing the right technology. So I think there's some connectivity that we can have and can bring to this through working with Patty, etc. So I'll finish there. thank you for listening. Just thank you again for inviting me. Hopefully there's some parallels and some experience with vascular gene delivery. I'm very happy to help and work with Patty and her team. Thank you.
We have a little bit of time for any questions anybody may have. What? Anything online? Well, thank you very much.
Pleasure.